Next up, we're going to hear about GDF15 in a very similar context from Carolyn. So thank you, Carolyn.
So I can start even if the slides are not up. So I'm gonna talk about GDF15 reactivity to acute psychosocial stress. And first, I'm going to explain why do we care about this question. So, as many of you know, you're there today probably because you're interested in mitochondrial psychobiology. Our hypothesis is that stress and psychosocial experience might modulate health through changes in mitochondrial biology. Also, variation in mitochondrial biology could influence mood and behaviors. And Cynthia showed this morning that different immune cells have different association with adversity and mood symptoms. I don't know if I should wait for this or no, but I'm going to keep talking. So there are many small studies showing association in mix of cells, mix of immune cells, PBMCs, showing an association between life experiences such as adversity, psychopathology, and mitochondrial biology. But we know that mitochondria are different between immune cells, so that's a major limitation. GDF15 represents a scalable biomarker to look at this association because it's the relationship between systemic communication. So more than 90% of the tissues in the body can express GDF15, but the receptors are only in the brain. So it's a clearer signal of mitochondrial energetic stress. Oh, my slides are there. How do I use this? Green button? Okay. So, yes, we think that GDF15 is a scalable marker of mitochondrial energetic stress. And I'm going to explain why, briefly, using the brain-body energy conservation model developed by Martin, Evan, and Alan. So according to this model, with aging, you have an accumulation of molecular damage that costs energy. And there is a finite energy budget the body can spend. So cells can send signal to the brain to tell the brain, we need to change something, we need to conserve energy. One of the signals the cells can send to the brain is GDF15. So as I mentioned, GDF15 is expressed systematically by the whole body to the brain to conserve energy. And GDF15 is also the protein that is the most upregulated with aging across multiple studies. It is the best marker of mitochondrial disease, so it's elevated in mitochondrial disease. GDF15 predicts disease risk and mortality, and in relation to psychosocial experiences, GDF15 has been shown to be elevated in psychiatric diseases. And in UKB, GDF15 was the protein the most strongly associated with social isolation. That was a study in more than 20,000 individuals. I did not include this here, but I have a poster after. So in UKB, I also found that baseline GDF15 predicts, so higher baseline GDF15 predict higher risk of psychiatric disease 15 years later. So in MiSBIE, we ask whether acute psychological stress can increase GDF15. I'm not going to go over the design again, but we used the TSST from MiSBIE with 8 time points in blood and saliva. First, I'm going to present the results for controls only. And for controls, we see that in plasma, there is a 5% increase of GDF15 10 minutes after stress. This was replicated in an independent study where we also see an acute increase in GDF15 that was not observed in the control condition, where participants had an IV in and blood was collected. But both in MiSBIE and in that study, we saw an increase over time across the protocol from 30 to 2 hours. And maybe it's related to the fatigue finding that Natalia showed before. Maybe people were bored, people are not used to not have their phone around for such a long time nowadays, so we need to understand better why we see an increase over the later point of the time course. Now let's turn to saliva. So in saliva we see even a bigger increase of GDF15 after stress, 5 minutes, so by more than 40% at 5 minutes. We also replicated that study in a larger study in collaboration with Robert Paul Juster. Here we saw about 30% increase 10 minutes after stress. So what about mitoD? So as expected, we found elevated GDF15 in mitoD in plasma. But we did not find acute reactivity in blood. But that doesn't mean that plasma levels in mitoD are not influenced by psychosocial experiences. These are results by Quinn Conklin. So she found that self-acceptance aspect of well-being was related to lower GDF15 levels in mitoD, while perceived stress and fatigue were associated with higher levels of GDF15 in mitoD, suggesting that mitochondrial energetic stress in mitoD can be modulated by mood and psychosocial experiences. So in saliva, that is really novel. We found that saliva GDF15 is elevated in mitoD. And here, mitoD participants also showed reactivity to acute stress. So we also find that there is a diurnal pattern of GDF15, and we found elevated levels at awakening in mitoD. Yeah, I want to go back and think about this. It's very important to say that the fact that we can measure saliva GDF15 and that it tracks with mitochondrial defects can be useful for future studies, not only for psychobiology, but for patients to track potentially energetic stress at home. Martin mentioned that health arises from the communication between different levels, and mitochondria help us adapt to stress, and when there is energetic stress, maybe we are less resilient, and that's something we can study by studying GDF15 potentially. Thank you.